Welcome back to the Biological Processes in the Brain lecture. This is our final lecture and it involves only Chapter 12, which is entitled Biological Processes in the Brain. In this chapter, we will be covering material that will most closely duplicate what you saw in an anatomy and physiology class. In fact, we will be primarily focused on the nervous system, in particular the brain or the central nervous system in general. But then we will have about a quarter of our time spent discussing the peripheral nervous system and a little bit of genetics and a little bit of hormones toward the end. And that will wrap up the entire course. To get started with in this particular section, we're going to start off with neuronal structure and function. And this is, again, fairly straightforward material. I presume most of you remember most of this material. The degree of detail that you need to know for the MCAT is fairly introductory. So there's not a lot of underlying biochemistry or genetics in this material. This material really holds along the lines of what typically shows up in a general psychology course or in a first semester course on the biological bases of behavior, both courses from the psychology department. So there's an overarching functional anatomical process, but not a lot of the underlying biochemical processes that's involved. For that reason, I'm going to skip through this material a little bit faster than I might if it were a general psychology course, because I'm pretty sure that you're relatively familiar with this material. Neurons. Neurons are the building block of your entire nervous system. Now, there are three different types of neurons, multipolar, unipolar, and bipolar, but the bipolar and unipolar cells don't show up in this material. So we only talk about your standard multipolar neuron, and we largely make pretend that it looks just like what you see on the screen. Multipolar neurons come in a variety of different sizes and shapes, but they all do have same characteristics, which is the dendrites all lead directly to the soma, and then there's one primary axonic branch that comes off, but that axonic branch may branch off into different directions. Remembering that the primary issue with respect to a neural cell is to manage the flow of action potentials through the electrical system of your nervous system. And that's the primary thing. So being able to sprout new axonic or dendritic branches and redirect electricity in different directions is one of its primary jobs. Now, neurons have three primary parts: the soma, the dendrite, and the axon. And those are the three that we're going to wind up focusing on in that particular order. In case you don't remember, the soma and cell body are synonyms. You could use one term or the other. They mean exactly the same thing. The soma is relevant for our purposes because it is the location in which your nucleus and all your genes are held in every one of these multipolar cells. Dendrites always bring information into the cell through tiny branches, as you see on the screen, and lead that information to the soma itself. And then the axon is the one primary branch away. Just to kind of give you a hint ahead of time as a possible reminder from what you've already seen, there really are three processes in terms of passing information through the nervous system. Those three processes are: one, there are charged particles that travel through the dendrite. Okay, there is no electricity in the dendrites themselves. It's simply the flow of charged particles. Secondly, there is an action potential, which is your unit of biological electricity that only travels down the axon. Okay, so from the dendrite to the axon, it's only operating in terms of charged particles. And so you go from charged particles in the dendrite through the soma, an action potential, which is biological electricity or action potential in the axon itself. And then the third unit of passing information is the neurochemical signal between cells, the psychopharmacology. For every cell that exists in your body that passes information through, you're going to go through those three processes. And that's why I don't say electricity travels through your nervous system. I've been saying information travels. I have to use the more abstracted term information just so I don't give you the impression that literally electricity follows an entire path. Electricity, only as biological electricity, exists in the axons and nowhere else in your nervous system. And that's an important thing to keep in mind, especially if you're going to be doing medical research, being able to distinguish where there is a neurochemical activity, a charged particle, an actual biological electricity is going to be important for the type of work that you do. Now, as I mentioned, we're going to cover the three parts of the cell in the order that they were shown on the last page. That starts us off with the soma. Why we get started with the soma first really is because we have to get the genetics out of the way, and then the dendrites and axons are both related to passing information through the nervous system itself, and so we kind of classify those two together. Now, the soma, again, is the same, has the same meaning as cell body. And it's relevant for us because it's the location where your nucleus is. And your nucleus is, of course, a spherical shape that has pores on it and contains all of your genes. And that's the primary purpose. So if you're wondering whether or not genes are relevant to human psychology, wonder no more, because genes are constantly used to manage human behavior, as well as the behavior of animals. It's part of just all of the processes that go on inside of your body. So there are many, many different cycles of biological activity that exist, and your genes are always being accessed for different reasons. Now, in terms of psychology, there are two particular reasons that your genes are accessed. And these are the only things that we're going to focus on in these lectures. And of course, this is a much smaller, myopic view of biology than you would get from a biology course, because a biology course is going to list an entire set of things in which genetics is going to be operating for. Our purposes in psychology come down to two things. One, your genes are going to generate hormones. All right, and then the hormones will have their effects, as you generally know hormones will, and we'll talk about hormones later toward the end of this entire lecture. And the second thing that genes do is they produce neurotransmitters. So if, for example, acetylcholine is a neurotransmitter in a synaptic gap, just like anything else, it's eventually over time going to get used up. The probability that the molecule will become destroyed or corrupted in some way will occur, and then that corrupted molecule will be discarded. So you'll have to make a new molecule of acetylcholine to replace it, and you always start that process off by sending a signal back to the nucleus to access the gene to make literally one more molecule of acetylcholine, which then has to get sent down the axon to the synaptic gap again. So for our purposes in psychology, again, two particular reasons why genes are necessary, and why why they are accessed all the time. One, to generate hormones, and secondly, to generate neurotransmitters. Now, as far as your genes go, genes are, of course, made out of chromosomes that generally look like the letter X, as you see on the screen here. And that's because the chromosomes are, of course, made from long strings of genes. So DNA, if you want to speak about this in a metaphorical sense, the actual double helix DNA would be like a very thin piece of thread that's very, very long. So if you try to imagine you got a piece of sewing thread that was 15 feet long, we could make pretend that that is a double helix strand. Very, very long, very, very skinny. Now, if you take the very end of that long string and start to fold it up, the length of the string will get shorter and shorter. And of course, the whole thing, by the time you finish folding it very carefully, will get thicker. So instead of having a very skinny piece of thread that's 15 feet long, you'll have something that's 1 inch long, but about 1.5 inches thick. You know, and that's kind of just the general way that I'm speaking about it, but that's essentially how your chromosomes are made. It is just made from a long string of DNA that's folded up very carefully, that becomes thick enough that you can start to see it under a microscope. And of course, with your naked eye, if you assume all the other material on it as well as just part of that chromosome. Now, you have a complete set of 23 chromosomes, and those chromosomes always come in pairs. So sometimes you hear you'll have 46 chromosomes, 23 from each parent. Sometimes you'll hear, like the company 23andMe, you're talking about you have 23 pairs. So sometimes people say 46, sometimes they say 23. Of course, you know the difference between the two. It's because chromosomes always come in pairs. Now, a chromosome itself is essentially tubular shaped, but there's always two of them. So you have two tubes right next to each other, and in the very center of those two tubes, at their, I'm going to say, waistline, they are cinched together by something called a centromere. And because the chromosomes are floating in fluid, you have these two flexible tubes tied together at the very center, which is what gives them that impression that they look like an X. And you can see that very easily in that image of chromosomes on the right-hand side of the page. But that's essentially the story of the layout of the chromosome. So when you look at a cell and you zoom in on it under a microscope, you can always find the soma easily because the soma is always the swollen section of the cell. And if you zoom in on that swollen section, you'll eventually see the nucleus, which is going to be a circle because you're looking at it from above and it's really a sphere. And if you zoom in further, you're going to wind up seeing 23 sets of chromosomes floating around inside of that nucleus. And then the nucleus that holds the genes are going to have to get those genes operating to produce the code to generate new molecules. And that's exactly what we're going to take a look at here in a brief, very nicely done three-dimensional animation of the operation of genes.
With computer animation, we can enter the cell to view this remarkable system at work. After entering the heart of the cell, we see the tightly wound strands of DNA, storehouses for the instructions necessary to build every protein in an organism. In a process known as transcription, a molecular machine first unwinds a section of the DNA helix to expose the genetic instructions needed to assemble a specific protein molecule. Another machine then copies these instructions to form a molecule known as messenger RNA. When transcription is complete, the slender RNA strand carries the genetic information through the nuclear pore complex, the gatekeeper for traffic in and out of the cell nucleus. The messenger RNA strand is directed to a two-part molecule called a ribosome. After attaching itself securely, the process of translation begins. Inside the ribosome, a molecular assembly line builds a specifically sequenced chain of amino acids. These amino acids are transported from other parts of the cell and then linked into chains often hundreds of units long. Their sequential arrangement determines the type of protein manufactured. When the chain is finished, it is moved from the ribosome to a barrel-shaped machine that helps fold it into the precise shape critical to its function. After the chain is folded into a protein, it is then released and shepherded by another molecular machine to the exact location where it is needed.
All right, that video I think can do a lot better job of explaining just how the process operates rather than through a very basic lecture. But you do get to see this process take place. The need for a signal coming from some location to make it back to the nucleus, fit through the pore, find the chromosome, unravel the DNA, get the code, move it out of the nucleus, and then generate the amino acid, and then get that amino acid and move it off to where it needs to go. Now, as I mentioned before, there are two primary purposes for the role of genetics in psychology. That amino acid that was eventually created is either going to be a hormone or a neurotransmitter. Those are really the two primary options. If it's a hormone, chances are it's going to be ejected from the cell. And the location where hormones are typically made generally is highly perfused in blood. So the cell in which that amino acid or hormone was actually created is likely literally next to a very small branch of a capillary. And so the hormone is ejected through the cell wall into the surrounding blood supply, and then it just washes away like a cork being thrown into a stream, and then it'll get where it goes based on statistical likelihood. Because obviously the molecule does not know where it needs to go. It's just a stupid chemical, and it'll go wherever it needs to go based off of chemical properties. So generally what will happen is in excess of hormones is generated, ejected into the blood supply, and then statistically some small amount of that will actually get where it needs to go, where the rest of it will just be floating around the blood supply or getting into your liver and kidneys and not doing anything that it actually needs to do based on the required function. The alternative is that the amino acid created inside the cell is a neurotransmitter, in which case that neurotransmitter is going to be taken through a separate biochemical process back down the axon, never down the dendrite, only the axon. It'll be taken down the axon to the very end, and then it will be placed in a vesicle, and it'll be used for when the cell fires later on. And if you don't remember how the process of neurochemistry works at the synapse, we're going to come back to that story again, but that's essentially what's happening. Those are the two primary purposes of the role of genes in psychology. Now that moves us on to the next slide here, which is about genes and cellular signaling. Genes are going to create hormones, as I've already mentioned. The hormones are going to get released from the cell into the surrounding blood supply. In most cases, when you're talking about hormonal activity, probably the thing that should jump out at you initially, your first thought is going to be your hypothalamus and your pituitary. And in fact, I'm sure you've heard of the hypothalamic pituitary axis. It's going to be the location for most of the hormones that you talk about related to psychology. Of course, hormones like adrenaline can come off of your kidneys, and there's hormones in your thyroid glands and other places around your body. Most likely, the hormones that you ever talk about regarding psychology are coming from the hypothalamic pituitary axis. You know, everybody's, of course, aware of adrenaline, but psychologists don't generally talk about adrenaline so much as a hormone, as they kind of treat it like a neurotransmitter, even though it gets, released into the blood supply and is functionally a hormone. It's just kind of the way psychologists talk about it. And I think the kinds of questions you'll wind up seeing on the MCAT are going to wind up following a very similar pattern based on how this course has been designed in the past and the way that your manual is written. I think that's very likely. This moves us on to the two functional components of the cell, the dendrites and the axons. Remember, I mentioned the dendrites and the axons are primarily important for the cell with respect to the movement of information through the nervous system. And of course, we're not saying electricity because only electricity exists in the axon, in the dendrites, and through the soma. It's the operation of charged particles. So the dendrites are carrying charged particles that are let in at the end of the dendrite. Now, why are charged particles let in at the end of the dendrite? Well, the entire process of the operation of a cell is a cycle. And we're just jumping in here with respect to the dendrite. So at the current time, you could just call it magical fairies. But for those of you that remember your anatomy and physiology, charged particles are let into the cell because of the preceding synaptic gap had some activity in it, and it caused, let's say, sodium, which has a positive charge on it, to come into the dendrite. But if you're not sure why, again, we're going to go through the entire process, and our discussion of the process will be a cycle. So we'll come back on the point where we started, and it'll all make sense in the end. So charged particles come into the dendrite, they float up the dendrite and reach the voluminous soma. When they reach the soma, they have a 1 millisecond chance, or 1 millisecond time frame, in which they have to produce a positive 20 millivolts worth of charged particles at that one time. So as you might imagine, there's a variety of different dendritic pathways into the soma, and any one of them randomly may be causing sodium, for example. I'm just going to stick with sodium because it's positively charged. Sodium comes up these dendritic areas, and if you ever reach a point in time in which a positive 20 millivolts of charged particles have reached the soma, then you will generate biological electricity in the axon. Otherwise, nothing will happen. So if you go backward in the circuit, and there was, an entire pathway of signaling that reaches the one cell that we're talking about, and at that moment that signal reaches the cell, it's not reaching the proper threshold of a positive 20 millivolts, then that signal has died, right? From wherever it had come from, it just cuts off at that point, and that's the end of that signal. This is, as you might imagine, why it's oftentimes important for pathways that are used repeatedly to generate more dendritic and axonic pathways along a general circuit to increase the amount of stimulation that occurs at every juncture to keep maintaining the flow of information through, which is essentially, in the most general sense, increasing the intensity of a signal through a particular pathway. Now the axon itself, if you've crossed that threshold, and make sure that you're aware of that threshold, it is a positive 20 millivolt signal within 1 millisecond. Okay, I say that again. The threshold to generate an action potential in the axon is that you must cross a positive 20 millivolt of charged particles within 1 millisecond period. If you do so, then you will generate a sequence of events where the cell membrane at the very beginning of the axon, an area called the Axon Hillock, will break down, and then the charged particles on the outside of the cell will start to rush into the cell itself. And that will start an entire sequence of processes that will generate a voltage differential which will run down the axon itself. And it's really the voltage differential which is what you refer to as biological electricity in this case. Biological electricity and the electricity that operates your laptop or a lamp in your house are completely different, right? Human-made electronic devices operate through a conga line of electrons, right? It's a string of electrons in a wire that just all move in one direction very slowly, but they all move in essentially a conga line. In terms of biological electricity, there is a rapid exchange of charged particles across a membrane that produces a differential of voltage, and that voltage runs down the axon. It's a completely different process. It's one of the reasons why you can't stick a light bulb in your ear and light it up. That's, of course, a joke. A hilarious, remarkable joke. Once your axon sends a signal, that action potential that travels through the axon is always going to be a positive 40 millivolt signal. Now that's as close to a constant as you're going to wind up finding in biology. I'm certain there are slight variations on it, but it's just said to be always positive 40 millivolt signal, and that's just the way that we're going to take it. So every time biological electricity, which is an action potential, gets sent down the axon, it's always a positive 40 millivolt signal and nothing else ever. So it's, I think, almost a guaranteed question that you'll wind up seeing on your exam. Now, one of the things about action potentials is they can have different speeds down the axon. Some move slowly, some move more rapidly. For some of you, you may remember the role of myelin, and we're going to mention myelin in a short period, but myelin coding on an axon will make the action potential move more rapidly. One other way to make an action potential move more quickly is to increase the diameter of the axon. But that little tidbit of information is not in your manual, so I doubt it's going to wind up on your exam. But it might be something that you're curious about anyway, given your background in biology. So a larger diameter axon will make a action potential travel more rapidly down the axon itself. Because the charged particles that generate the action potential either cross the threshold of positive 20 millivolts in 1 millisecond or it doesn't, the cell will be said to fire in an all or none process. The cell fires or it doesn't fire. When it fires, it produces a positive 40 millivolt signal, and there is no other possibility. So then there's no firing. And that's an important thing to keep in mind because at the heart of it, the type of information that's traveling through your nervous system is essentially an on-off signal. You either got a positive 40 millivolt signal or nothing. So, you know, I think as a modern person, your first thought is going to be, oh my God, binary code, Boolean operations. It's on or it's off. It's a 1 or a 0. And Yes, that is essentially true, although it's not going to work quite like a computer because the ones and zeros of a computer kind of string themselves together to produce code. You don't have that in the nervous system. What you do have is the rate at which these action potentials are coming down the pathway. And so you'll increase or decrease the amount of activity in the circuitry by how rapidly they're coming, right? Not the speed in, millimeters per second, but how frequently the cell is firing. That really becomes of great importance, because if a cell fires more frequently, more action potentials go down the axon, more electricity exists in the system, and you wind up seeing a higher level of activity in particular areas of the brain. And just as a quick aside to kind of connect the dots across all of these various lectures, that's what you're going to wind up seeing when you're doing an EEG or some other type of brain scan of an individual. If you're evaluating for the amount of activity that's occurring in a brain during an fMRI and somebody's doing, mathematical operations in their head, like counting backwards by 7, you're going to find certain areas that everybody's going to get all excited about and say, ooh, look, that area of the brain lit up. Why did it light up? Well, that area of the brain has more electricity in it. And you know that because there's more activity through those particular pathways. The increase in electrical activity requires more energy, so therefore there's more blood flow to that particular area. And so depending upon the brain scan technique you use, you're either measuring the changes in the magnetic properties of that area of the brain based on the increased blood flow, or like in the PET scan, greater radiation from that area because the glucose that's used as energy is more likely in that area of the brain because the glucose is being used as energy during the time that those cells are operating to produce the mathematical operations of the brain. And so if you kind of string these things together, you kind of get a better, more realistic sense of how your brain is operating. But as you might imagine, there is no one particular junction of your brain that qualifies to generate an entire intellectual sequence. It's just a hub of activity, right? It's like if you were following the electrical grid in South Florida, that runs electricity from a nuclear power plant to all of the residences and hospitals and so forth, and you find particular areas of the electrical grid that have more electricity than other areas, you don't point at that one area that has greater electricity, like a substation, and say, now I know why Joe turns on the lights. Now I know why Susan watches CNN on her TV. It's because of the electrical substation. Right? That kind of logic is just absolutely ridiculous. And it's just as ridiculous to point at an area of the brain and say, ooh, look, it lit up. Now we know where a person has mathematical operations in their brain. That's complete nonsense. You need a much larger grid of cells to work together in a unified manner to generate mathematical intellectual behavior or language or anything else you're actually looking for. So it's really been unfortunate since the 90s that people have pointed out the areas of the brain that light up as though that were really especially important. And it's not. You've got to hold some reality to this whole situation and remember that you're just looking at one particular section of the brain that has more electricity traveling through it at any one time, but that one area is not in and of itself some sort of calculator for a particular type of overt pattern of human behavior. That moves us on back from my rambling on about how to understand the brain to this section where we talk a little bit more about how the action potential actually operates. On the top right portion of the screen, there's a figure which is very important for you to pay attention to and grasp the understanding of how a action potential operates. The purple component in the center you probably recognize as a cell membrane because it has that bilipid layer. On the top of the figure is the outside of the cell, and on the bottom of the figure is the inside of the cell. On the outside of the cell, large boxes indicate high concentrations of a molecule, usually a charged particle in this case, and small boxes indicate a low concentration. So, as should not be a surprise to you now, sodium and chlorine are on the outside of the cell. The easy way to remember that is that life formed in the oceans. The ocean is salt water, and so it's not surprising that sodium and chlorine are on the outside of the cells. Inside the cell, you do, of course, see sodium and chlorine, but they are in low concentrations. What you see in terms of high concentrations are large anions. Those are the A- box over there on the left-hand side inside the cell. There are large anions which help maintain the inside of the cell to be negative. And you also have a large concentration of positively charged potassium. Now, what's going to happen in this case is that the cell membrane is going to have a variety of stressors on it that are trying to move these high concentrations into areas of low concentration, which is just standard diffusion from the field of chemistry. So in terms of diffusion, there is an attempt to move the distribution of large concentrations to areas of smaller concentrations so everything becomes equal. And then there's also electrostatic pressure, in which cases the charges that are equal are repelling each other and then the opposite charges are attracting each other. As I'm sure you know, but although I have not brought this up again, the resting state of a neural cell is minus 70 millivolts. And so that means that the inside of the cell has a negative voltage relative to the outside of the cell. Now that's when the cell is sitting idle and it's not being stimulated. Part of the reason why that cell has a minus 70 millivolt voltage relative to the outside is because of the large anions that you have on the inside of the cell. It's certainly not because there's a large concentration of potassium on the inside of the cell, right? You're talking about it has to be negative. So in this case, what you're going to wind up having, when you look at this particular image, is that with the inside of the cell being negative, the positively charged sodium on the outside of the cell is going to want to come in, because positively charged sodium on the outside is going to be attracted to the negatively charged inside of the cell. Plus, sodium is also going to want to come in because sodium is in high concentration on the outside and a low concentration on the inside. So between both diffusion and electrostatic pressure, sodium wants to come in. What about chlorine on the outside of the cell? It's in a large concentration. So the diffusion process of the concentration is going to make chlorine want to come into the cell as a result of fusion, but electrostatic pressure is going to keep it out because the inside of the cell is negative relative to the outside, and chlorine is a negatively charged ion. So the large concentration of chlorine on the outside is going to be repelled by the negative charge of the inside of the cell. If we go to the inside of the cell and we look at potassium, potassium wants to get the heck out of the cell because it's in a large concentration inside, and potassium is in a low concentration on the outside. So there's the process of diffusion that wants to get it out. However, potassium is positively charged, and therefore it's attracted to the inside of the cell because it is overall negatively charged, and therefore the process of electrostatic pressure is going to keep the potassium on the inside of the cell. The large anions, shown in the figure next to potassium, they don't travel through the membrane. They stay on the inside of the cell at all times, and we never even talk about them other than to just kind of refer to them as a background condition to help establish a negatively charged inside of the cell. Now, this whole process is going to cause a breakdown of the cell when you cross that threshold that we were talking about before a few slides ago. If you get enough charged particles up the dendrite into the soma so that you cross a positive 20 millivolts within 1 millisecond, you will cause a breakdown in the cell membrane of this cell. The breakdown of the membrane is going to start off by opening channels for sodium. And we're going to go through this in more detail in a slide or two, but what you're going to see is sodium, because it has two forces pushing it inside the cell, is going to come in, and then later on there will be some other flips and switches that occur which will cause potassium to exit the cell. The voltage of the cell will change, going from -70 at rest up to positive 40, and then the cell will reset itself. So the entire sequence of events requires some knowledge of where the ions are inside and outside, the concentration of them, the fact that the inside of the cell is generally negative relative to the outside, and the particular pores or pathways that the ions travel through in the cell membrane itself. Now, one of the things about the information being passed across the cell membrane is that once it occurs, you throw the cell into a changed electrical state. Instead of being at -70, the cell will travel up to positive 40 and then back down again to actually somewhere around a -75. Now, the cell has to reset its proper voltage before it can fire again. And that last portion of the sequence where the cell fixes itself and so that it can have the proper voltage to fire a second time is what we're actually going to see in this video here on the left hand side. And how the cell fixes itself is it initiates the activity of a sodium-potassium pump, because sodium has to be taken out of the cell and placed on the outside, and the potassium that had left the cell has to be brought back in. So this is a sodium-potassium pump that works against the diffusion gradient that occurs based upon the structure of the cell. And that's the issue. The sodium-potassium pump is going to require energy to pump the sodium out, to bring the potassium back in, and then only after this occurs will the cell be able to fire a second time.
The sodium-potassium pump is an active transport mechanism. 3 sodium ions bind to the protein channel, and an ATP provides the energy to change the shape of the channel that in turn drives the ions through the channel. One phosphate group from the ATP remains bound with the channel. The sodium ions are released on the other side of the membrane outside of the cell, and the new shape of the channel has a high affinity for potassium ions, and 2 of these ions now bind to the channel. This binding again causes a change in the shape of the protein channel, and this conformational change releases the phosphate group on the cytoplasm side. This release allows the channel to revert to its original shape, and as a result, the potassium ions are released inside the cell. In its original shape, the channel has a high affinity for sodium ions, and when these ions bind again, they initiate another cycle. The important characteristic of this pump is that both sodium and potassium ions are moving from areas of low concentration to areas of high concentration. That is to say, each ion is moving against its concentration gradient. This type of movement can only be achieved by the constant expenditure of ATP energy.
Now, that's a nice animation explaining how the whole process operates, the fact that it requires energy. In fact, when you hear about how much energy your brain uses relative to the rest of your body, a lot of it is actually related to the constant writing of the cells so that they can fix themselves and send another signal, because you are constantly using energy to operate the sodium-potassium pump. Now, in a broader sense, this whole sequence that I kind of talked about is laid out a lot more specifically here on the screen, and I believe this is very important for you to grasp. This diagram shows the processes that occur when a cell fires. And the entire sequence is something that you're going to have to commit to memory. You're very likely going to get questions about it, because this is kind of standard operating procedure in terms of cellular function relative to the field of psychology. So let's start all the way on the left-hand side, down by -70. When a cell is sitting and it's not being stimulated, or to say it more specifically, the charged particles coming through the dendrite into the soma are not crossing the threshold of positive 20 millivolts in 1 millisecond. Then the inside of the cell simply remains at -70. Once you do cross that threshold of positive 20 millivolts within 1 millisecond, then there will be a breakdown of the cell membrane at the axon hillock. The axon hillock is simply the first piece of axon just off of the soma. When that breakdown occurs, as we saw in the previous slide, the sodium channels are going to open up, and sodium has two reasons to come in, both diffusion and electrostatic pressure, which is going to bring sodium into the cell. And here at box number 1, you see the sodium channels open and the sodium begins to come into the cell. Now, this charge business, the charge of the cell, is a straightforward algebraic number line, up and down. Positively charged ions move you in the positive direction. Negatively charged ions move you in the negative direction on the number line. It's very simple and straightforward. If the inside of the cell is -70 relative to the outside, and you start having positively charged sodium come into the cell, then the inside of the cell is less negative. So it goes from -70 to -60, -50, -40, and so forth. And so the inside relative to the outside starts going up and is eventually going to reach positive 40 millivolts. Halfway there, somewhere around 0 voltages, the potassium channels are going to open. Now remember, potassium is in high concentration on the inside of the cell. So potassium starts to leave the cell. Now this gets a little bit confusing. The inside of the cell was negative. You start bringing in positively charged sodium, which is making the inside of the cell become more positive. Now the positively charged ions of potassium are leaving the cell, which is actually counteracting the role of sodium, right? Because sodium is positively charged coming in, but the potassium is positively charged going out. What's going to happen is the sodium channels are eventually going to close when you finally reach a positive 40 millivolts, all the way up at block number 3 at the very top of the parabola. And once you reach the top there, the sodium channels close, but the potassium channel is still open. And the potassium continues to escape from inside the cell. So if you have gotten to a positive 40 millivolt signal relative to the outside of the cell, but positively charged potassium ions are leaving the cell, then the inside the cell is going to start becoming more negative, right? Because that positively charged ion of potassium is leaving the cell. So you see here on this graph, the charge of the inside of the cell relative to the outside is going to start moving in the direction of negative. Eventually, the potassium will continue to leave the cell, and eventually the cell itself is going to return to its -70 level. But eventually too much potassium is going to exit the cell at the very end, causing a hyperpolarization. That hyperpolarization is important. This is not part of your textbook, but I give this to you anyway so to help you understand what its purpose is. If the end of the signal is hyperpolarized, that stops the signal from traveling backwards in the axon, because the trail of the action potential itself always has a hyperpolarized tail, which keeps the action potential moving in one direction and never splitting in half, and half of it going to the end of the axon and half going back up to the soma. So with that hyperpolarized section, this is where you start to get the sodium-potassium pump activated. Because you are at a hyperpolarized state, the sodium-potassium pump has to become active to bring the potassium back into the cell, which is against the gradient function, and the sodium has to be kicked out of the cell. And so as that operates for a brief period of time, approximately 1 millisecond, it will take care of that, because of course there are many sodium potassium pumps all over the surface of the axon itself, and that will correct the voltage of the cell itself, and then it'll be ready to fire. As I mentioned before, the issue with myelin helps make action potentials travel more rapidly through the axon. So that is at a faster speed as it travels down the axon. Not to occur more rapidly. The cell doesn't fire more rapidly, but the action potential itself travels more rapidly down the axon. How that operates is based upon the process you see on the screen here. When the cell initially starts to fire, as you see the depolarizing stimulus at the left-hand portion of the figure, that's like the Axon Hillock starting to fire just off of the soma. You have a full exchange of ions, as we were just talking about, that produce a positive 40 millivolt signal. And then that signal starts to travel toward the end of the axon, as I've mentioned, because it is trailed by a hyperpolarized portion, and it goes underneath of the myelin. But that signal is going to become weaker and weaker because there's no exchange of ions across the cell membrane when it's going underneath of that fatty myelin tissue. But myelin is designed in a way to leave tiny gaps so that the signal will reach the other end of a piece of myelin, and then there will be a full exchange of ions again, which will generate the full positive 40 millivolt signal, which will then travel on to the next piece of myelin. The reason why the action potential travels more rapidly through an axon under these conditions is because the signal does not have to wait for every single cell or for every single pore to open and close and for ion exchange to occur everywhere along the axon. Instead, you generate a full-size signal, and then it essentially travels underneath the myelin without having to activate every single pore, and so it can travel more rapidly that way. Please be aware that the spaces in between the myelin are known as the nodes of Rho-Va, or, if you want to call it Ron-Veer, that's fine, but I think you're safer speaking in public and saying the nodes of Rho-Va. And that is the space in between the sections of myelin. Now, when you finally get to the end of the axon, you're going to notice that the end of the axon always kind of opens up. It sort of looks like an elephant's foot. You got a leg and then this big stumpy thing on the bottom there. The end of an axon always has that big stumpy area to it. That's, the increase in size actually helps diffusion eliminate the action potential as it reaches the terminus of the axon itself. Now, what is going to happen that you've probably seen in biology class, but this almost never shows up in any kind of psychology textbook, is that there's an entire sequence of biochemical activity involving the release of calcium ions in the end of the axon. And we just almost never talk about these things, largely because they're not that relevant in terms of psychology. You know, either some of these underlying biological principles operate correctly, and then you have some kind of psychology to talk about, or the underlying biological principles don't operate, and then you either have a disorder or a dead person, right? Because the underlying biology doesn't work. So in many cases, you know, in terms of psychology, when you're talking about the biological principles underneath of it, either the biology works properly and then you can deal with the end result of behavior, or if the underlying biology doesn't work, you just don't have the behavior, and there's nothing to actually discuss in the end. So for our purposes here, when the action potential reaches the end of the axon, what's primarily going to happen is that the vesicles, which are made from the same thing as the cell membrane, a double lipid layer of fat, you're going to have each of those vesicles filled with neurotransmitter. Remember, the neurotransmitter was all generated by the genetics up in the nucleus and then sent down the axon through a process we're not going to bother to talk about here. And these filled fat bubbles, vesicles, are going to dock themselves in locations called docking stations once the action potential comes to the end of the axon, but it only happens when an action potential comes down. So when the vesicle attaches to a docking station, the docking station tears open the vesicle and it flings all of the neurotransmitter out into the synaptic gap. The one way to kind of view this is, if you and three of your friends were holding the corners of a square blanket, and in the center of the blanket you had a bunch of ping pong balls. So all of you yanked your edge of the blanket. The ping pong balls go flying in the air, right? Simple. Well, that's what's happening here in this artistic rendering of the end of the axon, except it's upside down. Right? Look at the vesicle. The vesicle is filled with ping pong balls. It attaches to the docking station. The docking station rips it open, and all the way on the right-hand side, what you're actually seeing is all of the neurotransmitters being physically flung out into the synaptic gap. And that's its process for moving over to the other side. It's just literally being tossed over in the direction of the next cell. And what is the next cell? Well, in most cases, and your material here really doesn't go into this detail, but it is most likely going to be referred to as a dendrite. You know, axons connect with dendrites, and then, as I've mentioned before, we have now completed the circle. Right? We started with the dendrite, and we talked about charged particles and through the soma and down the axon. And what happens when you reach the end of the axon? Well, you're at the dendrite in the next cell. And so we're starting all over again. So what's happening at this point is that the neurotransmitter is going to cross the synaptic gap, land on a receptor on the dendrite. The dendrite is going to open up a pore, let in sodium or some other charged particle, which will travel up the dendrite into the soma, maybe cross the threshold, generate an action potential, which will come down the axon, a release neurotransmitter, which will cross the synaptic gap, land on a receptor, open up a particle, a gate for the sodium, and then the sodium will travel up the dendrite back into the soma, and so on, and so on, and so on, and so on. Right? From the time that your nervous system is created at about 3 weeks after conception until the day you die. This process operates the entire time and never stops. So, from this particular slide, you're seeing the role of neurotransmitter being tossed out into the synaptic gap. And that's largely where we're going to kind of stop at this point in the story and move on to a new section, which I'm going to keep in this particular portion of the video, we're moving on now to the role of synaptic transmission. And as a reminder to you, even though your book was talking about the basic principles of a neuron and walked you through the activity of the dendrite and the soma and the axon, and now in a separate section is talking about synaptic transmission, I think it's probably better for you in general, just in terms of understanding biology, that the information that flows through your nervous system travels in three particular pieces. You first have charged particles through the dendrite and the soma, then you have biological electricity down the axon, and then you get synaptic transmission, chemical signaling between cells. You always have the sequence of those three events occur for every single cell. And the entire sequence of three has to occur over and over and over as information travels through your nervous system. And so here we start with the synaptic transmission story. Synaptic transmission, neurotransmission, they all mean, of course, the same thing. You're talking about the transmission of chemicals at the gap in between neurons. So you start to see a little bit of overlap here with what we just talked about. Vesicles filled with neurotransmitters are going to land on the docking stations. They're going to get ripped open. Their contents are going to get flung out into the gap that exists between the end of an axon and the beginning of the next cell, which is typically identified as a dendrite. You probably know from other classes that axons can butt up against other axons, and those are called axoaxonic synapses. In psychology classes, usually because the biology is kept at a relatively low level, you always kind of talk about a synaptic gap as axon on one side, dendrite on the other, and you generally don't talk about other kinds of combinations. So let's just keep it that way for our discussion here. One additional bit of terminology that I'm going to throw at you that I don't believe really shows up in the book so much is the terms presynaptic and postsynaptic. If the action potential always comes down the axon, and then there's a gap, and then there's a dendrite afterward, presynaptic means before the synapse, that's always the axon. Postsynaptic is always after the synapse, that's always the dendrite. So whenever you talk about presynaptic or postsynaptic activity, you're always talking about the axon or the dendrite, right, on either side of the synapse. And I know you're going to see that terminology when you do the A&P portions in the biology section of the MCAT. I'm hoping that, my comments now will kind of tie together some of this information, because there's a lot of terminology. There's many words that mean the same thing that get used in different ways. And hopefully for some of you, this may actually clear up a little bit of confusion based on the way science has kind of laid out the terminology and the descriptions here. Now, we're going to move on to this figure because this figure kind of helps us understand what's going on at the gap, the synaptic gap. Clearly, the top of this image is an axon, and you know that because you have the end of the axon looking kind of like an elephant's foot, right? The leg and the splayed out big foot on the bottom there. You also have the vesicles sitting around there that to me look like olives. Well, those vesicles, remember, the vesicles are made from the same thing as the cell membrane. It's just a lipid bilayer. And those vesicles only get attached to docking stations when calcium comes into the end of the axon as a result of the action potential reaching the end of the axon. Once those vesicles reach the docking stations, they throw their contents, which is the neurotransmitter, out into the gap toward the direction of the dendrite itself. At the dendrite is where you will have receptors, which are not really drawn here on this image very well. But what you have is A receptor, in most cases, being a large alpha protein that sticks out of the dendrite itself. So a large protein molecule sticks out of the dendrite, and that large protein molecule, like a big hair, is the receptor for the dendrite. And when the neurotransmitter crosses the gap, it will land on one of these large protein molecules, and the protein molecule receptor and the neurotransmitter have complementary shapes, like a hand and a glove, right? Opposite shapes that fit together. And what will happen is the combination of the neurotransmitter and the receptor itself will fit together so well that the complex of the two will twist. And the twisting action of that complex will cause a sodium channel to open. And it's typically sodium, although we'll see some alternatives. Psychology programs generally don't focus much on alternatives, at least in the lower level classes, but we'll see that on the next slide. But for example, as you see here, all the way on the right-hand side of this figure, sodium channels open up. So what's happening is the neurotransmitter lands on the receptor. They twist together. The twisting causes a channel to open that only sodium can travel through. Because we already know that sodium is in a high concentration outside the cell and is positively charged, and the inside of the cell is negatively charged, there's two chemical reasons for the sodium to rush into the cell immediately. It rushes into the cell, and then it travels along that skinny dendritic pathway up to the voluminous soma, where if enough of that positively charged sodium produces a positive 20 millivolt signal within 1 millisecond, you will then generate an action potential in the axon hillock itself, which will produce a positive 40 millivolt signal, which will travel down the axon. And when it reaches the end of the axon, calcium gets released at the end of the axon, causing the vesicles to get pushed against the docking station, which will get ripped open, and the neurotransmitter crosses the synaptic gap, and then the neurotransmitter lands on a receptor. And then when it lands on a receptor, it will open up a sodium channel, and then the sodium channel will let sodium into the cell for two reasons: diffusion and electrostatic pressure, and the entire process occurs again. And I am purposely repeating this because there's a whole sequence of events that always occurs the same way, but you have to understand the entire sequence of events or the whole thing just won't make sense. And, and just as a side note too, if for those of you that may be interested in doing work in pharmacology, it's that entire sequence where you're really interested in trying to figure out a way to get in there and monkey with stuff so that you could produce more neurotransmitters or modify the shape of the neurotransmitter or block a neurotransmitter. All of the biochemical pathways that exist inside the cell, including the genetic stuff, is really where you're going to wind up spending your entire career playing with information to try to figure out how you can actually get a change in the operation of the cell itself. Now, one of the last things that we're going to be talking about here is the charged particles. I have, up to this point, only referred to sodium. In a general psychology class, that's about the only thing I ever do talk about. If you get to a lower level biological basis of behavior class, you'll see a figure like this one. Sodium is a very common channel to allow sodium into the cell. And of course, when sodium comes into the cell, it makes this inside of the cell less negative, right? Because it's a positively charged sodium ion, so the inside of the cell becomes less negative. Therefore, it's called a depolarization or an excitatory postsynaptic potential. Why excitatory? Because when you depolarize the cell by bringing in positively charged ions, you move the cell in the direction of generating an action potential. That's why it's called an excitatory postsynaptic potential. Now, there are other channels, if you look at the next one to the right, where potassium will leave the cell. Why is potassium leaving the cell? Well, you got to remember from previous slides, I showed you that potassium is in a large concentration on the inside of the cell, right? And if that confuses you, just remember a very simple rule. Life started in the ocean. The ocean is made out of salt water, and so sodium and chlorine, that's the sodium chloride or salt, is on the outside of the cell. So that means potassium is in a high concentration on the inside of the cell. So there are channels where potassium will leave. What happens if potassium leaves a cell? Well, the inside of the cell is -70. You do have positively charged ions in there, which are potassium. Now, if you open up a potassium channel and you start letting the potassium leave, the inside of the cell actually becomes more negative. So now look at the name. That's called a hyperpolarization, or an inhibitory postsynaptic potential, IPSP. The inhibitory postsynaptic potential refers to the fact that the cell will have more difficulty reaching the point of an action potential simply because you have to go a greater voltage distance to reach the point at which it can fire. So if the cell sits at -70, you got to get a positive 20 millivolts for it to signal. But what if we allowed enough potassium to leave the cell so that the cell actually had a -80 inside voltage relative to the outside? Well, now you got to go positive 30 millivolts inside the soma before the cell will signal. The cell will generate an action potential, right? Because adding the positive 20 from the -70 takes you up to a -50 millivolts. And that's really the location of your threshold. So if you have made the inside of the cell more negative, you just have to add in more positive ions to be able to get back to that -50 location of where the true threshold actually is. If we go to the next channel, some neurotransmitters or locations in the nervous system will allow chlorine to come in. Chlorine is, of course, negatively charged, and making the inside of the cell more negative is, of course, an inhibitory postsynaptic potential, or, if you like, hyperpolarization. It's essentially doing the same job as having potassium leave, except they operate through different neurotransmitters. They exist in different areas of the brain. And as you might imagine, if you basically have the same process of hyperpolarization through two separate biochemical pathways, then there must be some other component that is relevant in this story. And of course there is. That story is not important for us. That goes too far afield. But if you're interested in psychopharmacology, those are just kind of some of the things that are going to have to be picked up by you over time. One of the last things that we see here is the role of second messengers. A neurotransmitter crossing a synaptic gap is really the first messenger. It's the first part of a sequence of messaging between cells. You almost never hear the term first messenger, though. You always hear about second messenger. The first messenger is just a neurotransmitter. The second messenger is, in this case, whenever the channel opens for calcium, which is outside the cell, which we really have not talked about, and calcium comes in, calcium may have different effects. In this particular case, it may influence enzymes which go to other receptor locations literally on the inside of the cell. That's an entirely separate receptor sequence that we're not going to talk about here, but that you would see in a pharmacology course. Or other types of effects that lead to second messenger effects, right? So the calcium comes in, then the calcium has a role in initiating some other activity, oftentimes which leads to the genetic activity inside the soma. So in many instances, second messenger systems ultimately affect genes. So calcium comes in at the dendrite, and then the second messenger system travels up the dendrite to the nucleus and gets into the nucleus, finds the chromosome it needs, unzips the gene, and yada, yada, yada. You're back to making proteins inside the cell itself. Of course, but never inside the nucleus. You always make your proteins outside the nucleus. One of the last things, actually the very last slide here for this portion of the lecture, refers again to the sequence of events of the interconnected cells. You have essentially the same diagram that we saw before, so there's a little bit of redundancy here in this case. It's giving you a basic time frame. You can see that it takes about 2.5 to 3 milliseconds overall from the point where a cell starts to initiate its action potential to the point where it's actually reset. So in this case, this figure shows that the action potential really is starting at about 2.5 milliseconds, but it's fine to shoot again at about 6.5 milliseconds. So in this case, it's drawn as about 4. Most of the estimates I've heard actually have been roughly about 3. So generally, I would say that if a cell was firing at its highest rate, it could fire at approximately 333 times per second. And if it were firing that quickly, that would probably indicate something like seizure activity. That's very, very rapid. Now, in a previous lecture, I know I have mentioned neurotransmitters and have showed you a table of neurotransmitters, keeping with the way your Princeton Review Manual has operated. There is again redundancy here. We are again looking at a table of neurotransmitters. This table is actually slightly different than the one that you saw before. What you're actually seeing in this case is a distinction between neurotransmitters that tend to be excitatory and other ones that tend to be inhibitory. Some of them are universally inhibitory or excitatory, like acetylcholine is universally excitatory. GABA is universally inhibitory. But histamine can be inhibitory or excitatory depending upon where in the nervous system it is and the surrounding biochemical environment to it. But these are simply kind of the subset of neurotransmitters that I've told you that are relevant with respect to the entirety of over 100 neurotransmitters that actually exist. Most neurotransmitters have very little to do with human psychology. These ones are at least peripherally related to behavior. Not even necessarily higher order thinking like mathematics and philosophy, but minimally motor behavior. Your ability to walk in a coordinated fashion or to dance or to go up or down steps involves some of these neurotransmitters like acetylcholine, for example. And disruptions in some of these processes can influence gross patterns of behavior. Again, not necessarily higher order thinking processes. If I had to guess, generally, most of your higher order thinking processes are actually being managed by norepinephrine, epinephrine, dopamine, glutamate, and serotonin for the most part. And that's a kind of a rough estimate story. That's not really something you need to worry about for the MCAT exam. It's kind of a little bit of tidbit of information that I'm kind of throwing at you regarding the role of neurotransmitters and higher order psychological functioning. It's actually limited to a small number of neurotransmitters altogether. All right, that will wrap us up for these 2 sections of this lecture on the brain, and then we will move on to our final sections afterward.